We're covering the nervous system and we're focusing on neuroglia or neuroglial cells, also known as glial cells. These are the supporting cells of the nervous system. In a previous video, we broke down the different types of cells and we talked about there's 4 types of neurons: anaxonal, bipolar, pseudounipolar, and then multipolar. And then we move on to the different types of glial or supporting cells, and we can divide them into the 2 different types of systems. Remember, we said the central nervous system is the brain and spinal cord. The peripheral nervous system is the nerves going to the periphery to talk to the receptors to bring all that information to the central nervous system so we can decide and sense what's going on and then have a reaction to it. When we look at the central nervous system, there's 4 types of neuroglia. In the peripheral nervous system, there's 2 types. So we're covering what they do and their basic function. A lot of texts just show graphics of these and not the actual cells. So we just focus more on the function of these. And I try to simplify them just to make them brief, just to have the basic understanding when you're going through the nervous system. I've been teaching anatomy for about 24 years now. Simplifying it for the nervous system is very difficult, and we try to put as much information together, but to simplify at the same time. I do have a study guide that goes over this as well that helps you to review. So when we look at the central nervous system, let's talk about the 4. The main ones, the most we see the most, are astrocytes. And what they do is called the BBB, or blood-brain barrier. In other words, we have to have a layer of cells to protect the nervous tissue. When we protect the brain and spinal cord, specifically the brain, we need to make sure that the blood is not exposing the nervous tissue to something that could disrupt the homeostatic balance of what it does. So it has a protective layer. The problem with that, though, if we're trying to introduce something, say like chemotherapy drugs, sometimes it's hard to learn the science to pass by this. So the blood-brain barrier is a protective layer to make sure we don't expose the brain tissue or the spinal cord tissue to something that would normally not interfere with it, but we need to make sure we don't override the balance of the tissue. It also regulates the different neurotransmitters that go through and pass through the different neurons. As a result, we can use this to control that interstitial environment. Remember, interstitial fluid is the fluid in between the cells. So we can regulate what's going in and out of the cells to make sure they function properly. When we look at oligodendrocytes or oligodendrocytes, they perform myelination of the axons in the central nervous system. Remember, myelin or myelin sheaths help to speed up that impulse, that action potential as we move along an axon. When we look at microglia, they help to clean up. So they remove cell debris, waste, pathogens, and that's within that interstitial environment. So as cells start to die off or they become older, we also get rid of the waste, or if we're exposed to anything like viruses and bacteria, they assist with that removal, mostly from a phagocytic standpoint, meaning they engulf and destroy them. And then lastly, ependymal cells in conjunction with the choroid plexus, they help to create that cerebrospinal fluid, or CSF. Remember, the brain and spinal cord is all one big continuous piece that's enveloped in meninges, and within those meninges we have cerebrospinal fluid. It helps with that mechanical protection, chemical protection, but also to create that continuous circulation of the fluid. So ependymal cells in conjunction with the choroid plexus that we'll talk about in a future video make cerebrospinal fluid. So these are all the supporting cells for the central nervous system. In the peripheral nervous system, what's analogous to Schwann cells are the oligodendrocytes. So Schwann cells are the ones that are doing myelination of axons in the peripheral nervous system, and then also help with some injury and repair. We do know that there is some repair done to neurons, but typically whatever neurons you start out with at birth is what you have. They're our longest living cells, and that's why we need to take care of them, because if any damage, say from a stroke or lack of blood supply, meaning oxygen, we can quickly destroy these, and it's harder for our body to heal relative to other types of tissue like epithelial tissue. So Schwann cells, myelination, again, that lipid appearance to give me the white matter, but we're doing this now for the nerves in the peripheral nervous system. Satellite cells are analogous to astrocytes and a little bit of microglia, but the satellite cells, to me, just like a satellite, they're moving around and helping to clean up the interstitial environment. So just like astrocytes, they're cleaning up around ganglia. If you remember from a previous video, the definition of a ganglia is a group of cell bodies. When we put a bunch of axons together, we get a nerve. So as we clean up around the cells, we are regulating the amount of neurotransmitters they're going through as we stimulate dendrites, and it goes through the cell body, the axon, when we look at an action potential running down a neuron, and then all the nutrient gases similar to astrocytes. So we clean up that interstitial environment. So we're trying to make these optimal for that chemical concentration, especially sodium, potassium, for that membrane potential as it moves along all of our nerve cells. So we have peripheral nervous system, 2 types, central nervous system, 4 types. As you understand these, it helps to guide you through the direction of the action potential and neuronal impulse, but first starting with just the basic functions of what are our neuroglia, or our supporting cells that we call glial cells.